All right. So we have reached a point where we had said we now clearly understand how the nerve controls muscle contraction with regards to getting calcium released from the sarcoplasmic reticulum. The particular region of the sarcoplasmic reticulum was known as the cisternae. Now, the next thing for us to really talk about is the power stroke. And we had said the power stroke is really describing the movement of one of those two myofibrils. Now, yes, everything is moving. We have stuff that's going to be sliding together and shifting over and all, but there's really only one of those proteins that is really reaching, grabbing, and pulling. So when we talk about the power stroke, we're talking about the myosin head and the change in the shape, the reaching, grabbing, and pulling by the myosin head. So the power stroke is describing that. And we had said, what's going on with that myosin head? It's a greedy bugger. It's reached out there. What happened is we had said ATP detached it from the myosin binding site on actin and caused it to reach forward because it wanted to grab and pull again. But what happened? There was no calcium. So it reached forward to grab and there was nothing to grab. So it's waiting. It's sitting there waiting. And then the calcium came along. And what did the calcium do? It combined with some of these other proteins that hopefully you've learned by now. We had said it combines with troponin. Now remember, on the surface of actin, we had tropomyosin covering the myosin binding site, and connected to tropomyosin was troponin. Well, as you know, the calcium comes along, combines with troponin, which pulls the tropomyosin off of the myosin binding site. And now this bugger can grab onto it and pull. And then it's stuck there. But not for long, because you have ATP that comes in and releases the cross bridge, and the myosin head reaches, grabs, and pulls again. Another ATP comes in, breaks the cross bridge. The myosin head reaches and grabs and pulls. And this is happening a multitude of times simultaneously with all of those different myosin heads. And what they're doing, you remember we have the sarcomere, and that's the Z line to Z line, and in the middle we have these myosins. And what do? They pull that sarcomere so it's shorter. And you've got a gajillion of these sarcomeres, all of which are getting shorter, and your muscle is contracted. That's how it works. Now, so as soon as you say, time to relax, I'm tired of flexing this muscle, what happens? Well, we have a protein called calsequestrin. And what does it do? It gathers all that calcium back up, puts it back into the cisternae of the sarcoplasmic reticulum. And so that myosin head is reaching and there's nothing to grab. It's waiting again. And so the calsequestrin is recovering the calcium, and that's what stopped the muscle contraction. So the muscle contraction will continue until it keeps reaching, grabbing, and pulling, grabbing and pulling. It keeps doing this until you no longer have ATP or calcium. So as long as you have energy present and you have calcium present, this process is going to continue. Now, ultimately, what had to happen to stop this process of reaching, grabbing, and pulling? We said we had to get rid of that neurotransmitter substance. We had said as soon as that neurotransmitter substance had come across the neuromuscular junction and combined with that receptor, it was causing a nerve impulse to enter into those T tubules and continue to release the calcium. So we had said in the synaptic cleft, we have an enzyme. It's there. It's something called acetylcholine esterase. Big name, but what it means is it breaks down the acetylcholine, stopping the nerve impulse, stopping muscle contraction. Yeah, it's as simple as that. Let's see what else we have to say here. And so we had said as the Z line is pulled in, as long as you've got energy, ATP and calcium there, those myosin heads are kind of like jogging in place. They're like reaching. They want to pull. They're workaholics. They're very energetic. So we had said that the muscle will contract until it is fully contracted when it's told to start contracting. Okay, so we had said as soon as you recover that calcium back into the sarcoplasmic reticulum, what happens? The troponin stops pulling on the tropomyosin, and so the tropomyosin covers up that binding site, so there's nothing for the myosin head to combine with on the actin. We all die. That's a weird transition, isn't it? But you know why I said it. It's because rigor mortis is what we came up next. I looked over here, and yet that's when a dead animal becomes stiff. And we had said that's easily understood because of decomposition. So as the muscles and as the cells are decomposing, that calcium that was stored in the sarcoplasmic reticulum cisternae, it leaks out. And that head, that myosin head that was sitting there ready to grab, it grabs and pulls. But there's no more ATP, so the cross bridge between the myosin head and the actin, it doesn't detach. It stays connected. And so that stiffness in the muscles is the myosin head's grabbing and pulling, and then they're stuck there. That's rigor mortis, and it results from a lack of ATP because you're dead, or the animal is dead. Let's not be personal. Yeah. Okay. All right, so I'm going to stop right there and let's see if we'll talk maybe a little bit more about energy.